name is Barbara Lech, and I'm a PhD student at the Wrocław University of Science and Technology. And I want to focus on RNA self-replication process, and I want to show you my results of my molecular dynamics investigation. It's quite opposite of what the previous speaker was talking about. I'm more focused on how the life could emerge from our planet by itself. So, as I think most of us heard about DNA replication. So it's a process which require a lot of enzymes, proteins to work, and it's required and essential for our life. However, I am more focused on RNA. And RNA replication, we can still find in some viruses and retroviruses. And there is RNA polymerase which catalyzes this process. We have initiation, then we can have elongation when the complementary strand is synthesized. And when everything is finished, there's termination, and we have a copy of our RNA. To maintain the process, the organism is using, for example, as in here shown, adenosine triphosphate as activated molecule for this process. However, I am focused on RNA self-replication, so formation of activated ribonucleotides into complementary strand, however, without any help of enzymes. And this is really important because RNA world hypothesis says that RNA could be one of the first molecules that complex, and that proteins were not emerged that complex like polymerase. So it had to for the survival, it had to self-replicate. And based on, for Jack Szostak and his team work, he says, or maybe his article says that with help of imidazolium-bridged nucleotides, the RNA self-replication process is real and it works. So why do I use molecular dynamics? Because all of the information we now have is mostly from X-ray crystallography. And I think most of us know that it's really expensive. It's for nucleic acid, it's really hard to create a proper crystal for this method. And the structure is fixed inside. So there is no insight about the dynamic of movement of the system. In here, you can see the results of investigation of guanine dinucleotide activated. It was on both sides of RNA, a double-stranded structure. Most importantly, it shows that this structure maintains this is canonical structure when there was some Watson-Crick interaction between nucleobases. However, from this X-ray crystallography, we cannot say the orientation of the imidazole inside in here. So this is where I come, and I use molecular dynamics because I can investigate many different structures, and as the name is, we can see the dynamic of the system. So I created 5 different systems with guanine activated dinucleotide, which is shown in here. So here is one guanine and the second, and here is this imidazolium bridge. Second system was created with inosine, and we chose that system because there's experimental results which says that changing of guanine into hypoxanthine, which is the nucleobase inside of inosine nucleotide, did not change the efficiency of the process. It maintained the same rate and fidelity. Secondly, I also created imidazolium-bridged adenine nucleotide, which we know from the experiments that this complementary uracil, it's really hard for the system to replicate. It had really low fidelity and rate. And at the end, I created 2 more systems where adenine nucleotide, it was almost the same, but in the complementary strand, I changed uracil to thio-uracil. And then I made 1 more where the imidazolium-bridged dinucleotide was to thio-uracil. And that was also the case that experiments showed that change of uracil to thio-uracil make the system more stable, and the rate was higher. For those who don't know this nucleobases, this is hypoxanthine. If in here was NH2 group, it will be guanine. So it's really similar. However, it can only create 2 Watson-Crick hydrogen bonds with a complementary strand. And this is thio-uracil. So in the second place, it's supposed to be oxygen in uracil. So in here is sulfur atom. My methodology. So as I already said, I use molecular dynamics. I perform simulations in amber param-oil-free force-fed. Each system was closed in the tractolated octahedral water box with 0.15 moles of salt. And each simulation was held in 300 kelvins and 1 bar. And for each of the 5 systems, I gained 1.5 microseconds of simulation. And at the end, I used RMSD clustering, dbScan, and Chymine to better understand the results. So dbScan is something like machine learning. I gave it some parameters like epsilon, as you can see in here, and it was searching and creating clusters with similar structures. And Chymine is quite easier to understand. I just told to the program that I want 10 clusters, and it created give me 10 clusters with similar structures to itself. So the results. In here is the first system with activated guanine nucleotide. It shows that it shows results similar to what Jack Szostak get from his X-ray crystallography. It's beautiful canonical structure where Watson-Crick interactions between nucleobases are created and stable for the whole simulation. On the other hand, with the it's supposed to be in here. So it's activated inosine nucleotide. For most of the simulation, it maintained this canonical structure with Watson-Crick interactions. However, it isn't as stable as with guanine nucleotide. And as you can see in here, there is no canonical structure. We can say it's all break. But quite interesting thing is that this is this activated dinucleotide, and I call it that it looked like G-letter structure. So it's quite interesting because these 2 nucleobases are stacking. So they're one on top of another, and it cannot any longer maintain standard canonical interactions. Most interesting thing is that I confirmed that with activated adenine nucleotide, there's no canonical structure at all. However, this G-letter structure also occurred. So that this result is comparable with the experimental data, and this G-letter structure is quite interesting that it also occurred in another system. For me, one of the most important results which I get is with change of uracil to thio-uracil in the complementary strand. Because as you can see, in this case, I get clusters where the canonical structure was created, and there are Watson-Crick interaction between nucleobases, complementary nucleobases. So it's improvement because in adenine activated nucleotide with complementary uracil, there was no standard interaction. And as you can see, the structure doesn't look as supposed to. However, it wasn't like 50% now of the structure was maintained in this look. However, in this case, also adenine dinucleotides created this G letter structure. And the last system, so when I activated thio-uracil, the dinucleotide, I'm sorry. So in this case, something really interesting is happening because just the dinucleotide is just diffusing away from the binding site. And still, as you can see, for example, in here can maintain canonical interactions between nucleobases like or in here, but it ends up somewhere in the middle of the double-stranded RNA structure. And that was all of my results, which I wanted to show you. So in summary, inosine dinucleotides exhibit weaker Watson-Crick interactions comparable with guanine dinucleotide, which is quite surprising when we have experimental data which says that it's supposed to be the same. Secondly, I show you that this imidazolium-bridged nucleotides created canonical stack structures, and this G letter structure which I want to investigate more in the future. Second, and the last thing, or more, almost last thing, is that change of uracil to thio-uracil in the complementary strand make the system more stable, and that's proof why the fidelity and rate of this process was higher. And the last thing that I already told, that imidazolium-bridged to thio-uridine dinucleotide just diffuse away from the binding site, and that could be the reason that it's at the end of the RNA double helix. Maybe when it would be in the middle, it wouldn't diffuse away. However, I will try to investigate that. And I want to just thank you for the presentation. And that's all.